Organic Chemistry, Lecture 8, Nitrogen and Phosphorus Containing Compounds. This lecture is going to be divided into two portions primarily. The first being amino acids and their respective peptides and proteins, and the second portion being phosphorus containing compounds. We'll start with the former. Amino acid structure is going to be very important when it comes to the MCAT, because obviously amino acids are the building blocks for every single protein. They contain an amino group, a carboxylic acid, and an R group. The properties of these amino acids vary based on the R group that's attached. Amino acids are interesting because they are amphoteric, which means that they form Zwitter ions. And they form bonds via hydrolysis and dehydration reactions, or condensation and dehydration reactions. If you don't remember what a Zwitter ion looks like, it looks something like this. The picture on the right is in a neutral solution, but when it forms a Zwitter ion, it's going to have charge present. So, if you don't remember what a Zwitter ion is, it's a compound that has both a positive and a negative charge present. Here are the 20 fundamental amino acids that you will likely see on the MCAT. As you can see, there's different ways to group these, and the groupings are listed below. I would strongly encourage you to have these memorized, along with their shorthands. Now that we've talked about some fundamentals of amino acids, let's actually go into the synthesis of these compounds. Starting first with the Strecker synthesis. This one starts out with an aldehyde and works its way through. The first step, as you can see, leads to the formation of an aminonitrile, which is the last compound in the chain. This then later feeds into part 2. This is where the aminonitrile then is converted into an amino acid proper. The next synthesis we're going to go into is the Gabriel synthesis, or as it is also known, the malonic ester synthesis. This is a pretty intricate looking synthesis, but really the dead giveaway when it comes to the MCAT is when you see potassium phthalimide, which is this compound right here, and some sort of halogenated malonate. If you see these two, you will likely be looking at some sort of Gabriel synthesis, which is going to help you in determining what's going to happen in the reaction. If you are to see these two compounds, just know that the end result is going to be, quite simply, an amino acid. If you want to follow the steps through, you'll notice that the key reaction is the SN2 reaction in step 3. This is going to add the R group necessary. Then, with the addition of base and acid, you will eventually create the amino acid proper. Note, the Strecker synthesis and the Gabriel synthesis are the two most complicated syntheses you will likely see on the MCAT, so it would be in your best interest to spend some time going over these and understanding what mechanisms are going on. Now that you've covered nitrogen-containing compounds, let's go into the second portion, the phosphorus-containing compounds. In terms of physical properties, the most common are phosphate groups and inorganic phosphates. These are often associated with energy and cellular machinery. Listed at the bottom right is ATP, which is what every single biological organism uses for energy. As you cut phosphates off of ATP, the energy output gets less and less. So when you go from adenosine triphosphate to adenosine diphosphate, you get a pretty substantial energy payout. But when you go from adenosine diphosphate to adenosine monophosphate, you'll see a substantially lower payout. Being able to recognize these phosphates and how they behave in organic systems is going to be very important. In terms of acids, phosphoric acid is very unique because it's one of the few acids in organic systems that is triprotic. Because of this, it is often used in biological systems when multiple donations are required. Phosphodiester bonds are interactions where there is a phosphate in between two bases. This is generally found between DNA and RNA bases, which are listed below. This bonding structure is pretty characteristic of only nucleic acids, so if you see these, you can generally assume that it has to do with either DNA or RNA. That covers our portion of the lecture on phosphodiester bonds. As you can see, those, along with nitrogen-containing bonds, are very crucial to organic chemistry and biochemistry as a whole. With that, we will conclude our discussions of functional groups and reactions and syntheses regarding these groups. In the next two lectures, we're going to go into more of an instrumentation and analysis portion of organic compounds. So join us in lecture 9, the next one, which is covering spectroscopy.